killing the cells, correct? They are killing the cells. In addition to cancer cells, they are killing to your normal cells also. That's why they are very, these molecules are showing various kind of side effects, because specificity is very less for the cancers. So you have to perform the safety study also. You have to check the specificity of your product. All these things you have to do in discovery and development phases. Like I have mentioned the first phase. So here all these studies we have to perform. Okay. So, but what you for this study you need molecules, a single molecule, okay, or any API. If any reagent, suppose if you synthesize, that is called hit molecule. If it is showing good activity, no? That is called hit molecules. When you perform some study, when it is showing very good activity, toxicity wise, everything is good, that become a lead. Okay. So we have to screen many compounds, like around 10-20 compounds. They are showing very good activity among the 100 or 200 compounds. Because all compounds are not active. All compounds are not very good. Some of the molecules are very good in activity, but their toxicity is also high. So that kind of molecule we have to discard. Then after having all these things, like in vivo study, we have to perform with animal model. So if all one of the whatever the molecule you have selected, if you qualify all the parameters, okay, from this, then it becomes your active pharmaceutical ingredient. That means it is ready to, now you can go for the next trial. Okay. So so now the main challenges is now you have performed 50 gram or 100 gram scale in your laboratory. That is feasible. But if you need more quantity, what you will do? You have to go for the bulk scale batches, like kilogram scale, 36 gram, 50 gram, 100 gram, 500 gram scale also. Okay, so when you need any molecules in the bulk, then what we have to do, we have to go for the plant scale batches. So going in plant scale batches in industry is not easy. Obviously, you are doing experiment. Your discovery is from your laboratory, a small lab. You are making a molecule. You are checking the molecule, its feasibility, whether we can synthesize it or not. Okay. So, and you have tested that your compound also. This is very good active compound. Activity is very good, and you have performed the enzymatic assay. Everything is over in this stage. Okay. So process development, I did not study in the books that is there any step for process development drug discovery. I did not see that. So my main aim was to bring this topic. This become a interest of presentation, because people should aware about how much struggle we are doing for the development of one molecules. We have to go for the bulk scale synthesis. So lab scale batches, we are also doing lab scale batches, but we are doing lab scale batches in the with the view of this large scale batches. Like we have to do all the negative experiments. We have to check the rates in that with a different temperature, because whatever you are doing in the laboratory is completely different from the plant batches. It's not like that you have done the molecule and you have made this, so it will also make, you can make this one in the bulk scale batches. It never happened actually. So because small scale batches, when you are doing, there is no risk at that. The risk is very low. If your batch got failed, also no problem. We have spent around 5,000, 10,000 rupees for a single reaction. So 10,000, 5,000, okay, that much loss the company can bear. But if you are going for the plant batch, you have to take 1 kg scale. You just assume that we are spending for one reaction, we are spending around, sometimes we are spending 20 lakhs, 30 lakhs rupees also, or 50 lakhs. And the most of the raw material cost is going to crores of rupees also. So that time it is very risky to perform any batches in the process development, like in a plant, because that is 40, sometimes 14 kg, 15 kg batches also we have to perform. So before going to the plant batches, we have to do several kind of experiment. We have to prove ourselves. We have to we have to generate several documents like batch manufacturing record, BMR, like those BMR, TTD, all these documents we have to prepare before going to the plant. Then only this is acceptable. Okay, your process is set. Now you can go for the plant batches. Because you people also have performed several experiments and synthesized molecule in your laboratory. Simply, you have added this one, added this raw material 1, raw material 2, and heat it for 50 degrees Celsius for 8 hours, then filter it. Like that, you people have made. But you never check the purity of your compound. Some solid came, okay, this is very good. Solid came, now my product is ready. We will check the activity now. So it's not like that. Here we have to check so many things like first how much conversion is there, how much starting material is present in your reaction mass. Okay, so these things you have to check from first of all. You should check the reaction with TLC, and TLC also it's not appropriate, because if your starting material is there for 1 or 2%, it will not be visible in TLC. You have to go for HPLC data. From HPLC data, you will come to know how much percentage is there, 1%, 2%, 3%. That also matters in process development. You don't think that, okay, 1% is there now, 99% converted. It's not like that. It should be under the limit, and it should not present in the next stage. Otherwise, it will carry forward. I will explain in the next slide how it takes place. So for preliminary examination, it's very good. You can do that, and you have to fix the specification limit. Like if you will perform several kind of reactions, you will come to know, yes, we can make up to this limit. We cannot purify more than this percentage. So you and your client, whatever the company who is going to develop the molecules, have to discuss with them, and you have to fix the specification limit also. Then you come to the process optimization. Process, before going for process, you have to study several kind of study you have to perform, and you have to optimize the process. You have to do all the negative experiment. So that also I will tell you how, with examples, how we are doing the process optimization. Then we have to, after doing all this experiment optimization, then we have to write a process write-up. Process write-up means it should be very easy. Okay, it should be because in plant scientists are not working. Some casuals are also working. One of the chemical engineer will be there, and so they don't know what is this, what you are doing. Okay, for them this is just an experiment. Okay, this one process write-up it is written, so we have to add this, we have to add that raw material.
Okay, right.
Okay. So in that way you have to write the process write-up so that everybody can understand. Then based on this process write-up, whatever you have given, they will generate one batch manufacturing report, BMR. After that, they will make your TTD, that is called technology transfer document. Then they will start your reaction. Okay. So from here you can see that very small reactor or RB they are using in the lab. But here if you come to know 100 liter, 200 liter, 4,000 liter, 6,000 liter reactors are also available in the pilot plant stage. So when we go from the small scale synthesis and to the large scale synthesis, what challenges we are facing, what are the issues are coming, that I am going to discuss. Like first and very common issue is unexpected impurity, because unexpected impurity may come because of the variation of temperature. If you are taking a small batch in a RP, if you are adding some other reagent like any other reagent, suppose you are doing the reduction reaction, you have to some of the molecules you have taken, keto molecules, and you have to reduce it. You have to add sodium borohydrate. Slowly you are adding. So the temperature may range up to how much percent it will range? 5 degree, 10 degree. That is not very dangerous. But if you are going to add 1 kg of sodium borohydrate into the reactor, you just assume that there may be chance to raise the temperature up to 60 degrees, 70 degrees Celsius also. So that time what will happen? Whatever the raw material is there, so that may degrade, or product which formed, that may also degrade. Okay, so this impurity is very common in the process development. Always we are facing whenever we are going for the plant batches, this problem is common. So you should know, you should be ready actually. If problem comes, you have done a very excellent job in your laboratory, but sometimes it doesn't work in the pilot plant. So, and exposure of moisture is also more when we are doing in the bulk scale batches. So this impurities are very common, unexpected impurity. It comes, generally it comes. And the problems with raw materials. Sometimes whatever the volume you are using in the lab and volume you are using in the reactor is different. Okay, so sometimes solubility issues also comes. Some of the raw materials they got dissolved very easily in the lab scale batches, but in plant scale batches it's not dissolved. We have to increase the solvent volume or something we have to do there. And raw materials also we can see that vendor to vendor also there are some problems. Some of them are giving good raw materials, some of them are giving not good, some China material also there. So their quality and other vendor quality that may vary. That also impact your reaction. And solvent also. In solvent, we don't know while you are using in the lab. Very simply, your THFS you add it and do everything. But here, if you are going for the plant batches, you have to check everything. Like acetone, if you are using, you have to check the moisture content. THF, you are using moisture content, you check. Because if moisture is there, that may also impact your reaction, and that may generate your impurity in the reaction. So it's a very common problem. So you have to solve, you have to take care before going to the plant scale. What we have to do. Then some of the steps. Like recently we are doing one project where up to 25 gram, 50 gram the reaction is going very good. When we are going for the 100 gram scale, the reaction is not going to perform well. Okay, so what we have to do if it is not scalable, if it is not going for the bulk scale, and that molecule is very active and that is also under clinical trial also. So tox studies are going on. Some of the experiments, some of the reactions are not scalable also. So for that, what we have to do, we have to change the synthetic route. We have to change the route. This route is not working, so what we can do the next. So we should be ready to make other route also. Okay, and and in production, for scale up batches of process chemistry, we need more time. Time means, suppose if you have to charge the solvent into the reactor, it itself take half an hour. That half an hour you can complete your reaction in your lab. Okay, so we are not that much fast in the plant. We have to go slowly. Addition also we have to do slowly, because if I will add, okay, if I will add for very fast, what will happen? The temperature may rise suddenly. Okay, if you are using suppose lithium aluminum hydride or penemetal lithium kind of reagent, any pyrophoric reagent, so that kind of reagents are very dangerous. Okay, so that may catch fire also, and if it catch fire, it's a very difficult to control that kind of fire. So we have to be very careful, because we should be ready. We should be always ready. It will take longer time. For that, we have to perform stability studies. You do the reaction for the longer reaction type at this particular temperature. If temperature may reach, what will happen? Which kind of impurities are generating? So these kind of experiments are very common. We have to perform. Okay. So, and wrong temperature. Wrong temperature means, suppose if your reaction is at 50 degrees Celsius. Okay, and you have already, okay, 50 degrees Celsius is there, we can go for the 50 degrees Celsius reaction. You have set the temperature of the jacket, and now you forgot it. Okay, then sometimes what happens, because of the reagent, because of reaction, the internal temperature may also change. So wrong temperature also, it can create a very big problem. And other parameters like moisture content, order of addition, filtration methods, these are very important. Like filtration also, if you see filtration methods, some of the solids are very fine, that may pass through the Nudge filter also. So those kind of problems we have to be very careful. We have to check everything before going to the pilot plant scale. Order of addition is also important, okay, because if you will add very fast, obviously 100% some problem will come. So your order of addition, like if 1 ml per minute means 1 ml per minute it should go, and you have to check all of the temperature and all the reaction parameters you have to check. You have to be there when we are performing any batches. Now I will give you some examples like how we are facing the problem in the process chemistry. I will give you the real example here. Like one of the molecules there, like diketopic derivatives. Okay, I will not disclose the name of this molecule, but you can see the structure and you can just see it's very one of the simplest scheme I am showing here so that you can understand what kind of problems are there, how they solve the problem. So when this is simply it's a L-lysine molecule, lysine is amino acid. Amino acid, this is the intramolecular addition and form this diketopic ring. Okay, actually our final molecule is this one. Okay, this diketopic ring. But to synthesize this, we have to perform these 3-4 stages. Okay, once you will synthesize all these stages, then only you can go for this final molecule. So for that purpose, we need one starting material, that is L-lysine, which we have to protect from this N-hydroxysuccinamide. Okay, the reason to protect this N-hydroxysuccinamide, it's a very good living group, so that it can easily attack this electrophilic site and can cyclize. But I will show you next slide one by one what problem we faced in every step. So you can make this diketopic derivatives, and simply you can reduce this palladium carbon under the presence of hydrogen, and you can cleave it. So the free amine you can get, which can be easily coupled with this fumaryl chloride, and you can get this amide linkage. After getting this ester, you just see this ester, you can hydrolyze it with sodium hydroxide to get the acid. And this acid we can simply make sodium salt in the presence of sodium hydroxide. So by looking at this scheme, it's a very simple scheme of what is there in this scheme. Nothing. It's very simple. So in laboratory, we also perform this reaction up to 100 gram scale. That reaction was very simple. Everything was perfect. But when we move to the pilot plant, when we move for the kg scale, like any step 1. Okay, let's step 1. What problem we faced, this we end up with some impurities, and that was also in kg scale. So then what was the impurity? So first stage, what happened? This molecule, if you will add trifluoroacetic acid, this BOC will go. This is a simple protecting group of amine. So this BOC will go. Now it is free to react. It will react with electrophilic site. Okay. So first one it will react. Okay. So it forms this amide here. But second reaction, this intramolecular addition could not take place. Why? Because this hydroxysuccinamide was cleaved. Okay, so once it will cleave, you will end up with acid. Okay. Then we thought, why this reaction not done? Then we have to do all the studies, what happened in that. Then we found that in the LCMS we have done, we have found that we are getting this compound. So once you will get this one, it will not cyclize. That will end up with, you will not get your desired product. Or if you get the desired product, means that yield will be very poor. So other one, what we got, this impurity also we got. That is simply L-lysine we got. This severe protected L-lysine we got. So both of these impurity we observed when we are doing the plant scale batches. So what cause is there? Then root and cause we found that it may happen because of the moisture. In that solvent, like we are using TFA, then it's dichloromethane. After that, what we are doing distillation. Okay, so in distillation also, like to remove the trifluoroacetic, we need higher temperature. So higher temperature means whether this is stable or not. It was stable, but for certain time only. After around if you do it 15 minutes, 20 minutes, that is fine. Up to 1 hour also no problem. But if it is taking 6 hours or 5 hours for this distillation, that may also cleave. So we found that this problem comes because of high temperature for longer reaction time, for longer distillation time, and to remove the moisture, we introduce this trifluoroacetic anhydride. So what it will do, if you will add into the reaction trifluoroacetic anhydride, it will cleave into trifluoroacetic acid only. Okay, so there is no problem. It will quench your moisture present in the reaction. So for that we have introduced this trifluoroacetic acid. Somehow we solve this problem. We have added TFA, we use dry solvent and distillation of TFA at low temperature. When we went for that, we got very good yield. But this reaction, same reaction we performed in the laboratory, there was no problem. But it came to the plant in 6 hour, 8 hour distillation time only spoiled the reaction. Okay. So this kind of problems comes. So we have to be always ready for to solve the problem. Then coming to the step 1. Now we have synthesized this step 1 with this NH immediate with diketopiperazine scaffold. Then this reaction is autoclave reaction actually. Autoclave reaction means there is a reactor where you have to pack everything and you have to give the pressure at very high pressure, means around 8 kg, 10 kg presses you have to give. Okay, so first problem, this palladium carbon that may catch fire very easily. Okay, so while addition only you have to be very, very careful. Okay, then hydrogenation reaction. This reaction went very well. So that reaction was in acetic acid. The acetic acid we are using as a solvent we are using in this. So we are end up when it will cleave and there is base and acid. Obviously it will form salt. So it forms acetate salt. But the problem what happened, this acetate salt was hygroscopic. When you are doing small batches, you are doing fatafat, 5-minute, 10-minute reaction, so there was no problem. But when we go for the plant scale, that is taking 3 hours, 4 hours for the filtration. So that time we observed that this molecule was very hygroscopic. So if the molecule is hygroscopic, it's very difficult to isolate, it's very difficult to dry the molecule. Okay. So we have interchanged the salt by addition of concentrated HCl. So this process worked nicely, and we got, instead of the acetate salt, we got chloride salt. So after the completion of reaction, we have to distill the solvent like acetic acid. We have to distill and we have to remove the acetic acid under the vacuum at 60 to 65 degrees Celsius. That time again one problem was coming. We are getting this N-acetyl impurity. Okay. So one of your molecule like amine was getting acetylated in the presence of acetic acid. So that problem also we faced. So to remove that N-acetyl impurity, this concentrated HCl was working fine because you can reprotect this N-acetyl with the acid. Okay, so that this kind of impurity we are generally analyzing with LC mass and HPLC. Now coming to the stage 3, once you successfully made this molecule, you have to perform this third stage 3. Like you have to do the amide coupling here. Okay. So if here also, if you are thinking that okay, we will take this impurity and we will purify in the next stage, that will also create a lot of problem. That will carry forward to the next step. That's why you have to purify your intermediates under the specification limit. Otherwise, you will face some problem. Without purifying, without developing a proper process, you cannot make your molecule nicely. Okay, with specification limit. So this amine we are taking. First, even small trials we have done by taking this N-acetyl impurity. So we got the molecules along with impurity. N-acetyl impurity. You can see that this is the mass of our desired molecule and this is the mass of our N-acetyl impurity. Because we have used other molecule, this stage 2 along with N-acetyl impurity. So we end up with this impurity also. So you should not use any of the impure molecule for the next step also. Then other here also 2 stages are there, like sodium hydroxide stage for hydrolysis. Here what the problem we are facing, if we are using high more excess of base, this amide bond was hydrolyzing, like we will end up with this open molecule. Okay. So because amide is also susceptible to base and acid, that may cleave very easily. So you have to be very careful when you are using any base. So here also, like we got this we just expected this 2 impurity may form because mass is same. If you see the 470 and 470, both the impurities mass are same. So other possibility we thought that may reduce the influence of acid. Okay. So among these two, one of the impurity form. Then for hydrolysis, we are using generally protic solvent like methanol water. So we also perform some of the experiment in methanol water. We are ended with this acetyl impurity here, methoxy impurity. So we change the solvent. We change for the other solvents like tetrahydrofurans for the hydrolysis of the mixer. So like that we should select the solvent, we should select the reaction condition, we should select the reagent or base, acid, anything. By seeing all these critical steps. Okay. Now this step 4 was also very simple. It looks very simple. So everybody told, okay, what is there? This is the salt formation, but it's not easy. Salt formation means the reaction was very heterogeneous here. Because water, if you make any organic compound as salt, that will be water soluble. Once it will solubilize in water, how you will isolate it? It will be very difficult to isolate the molecule. Okay, freeze drying you can use, but for what you will do for kg scale batches or big scale batches, so that was not feasible for the plant scale batches. So we have performed in the methanolic sodium hydroxide we have prepared, and we performed this reaction. The plant batches reaction was very nice. We are getting the product, everything was perfect. But in plant batches, the problem, the reaction was heterogeneous. It was difficult to identify whether reaction got complete or not. Because in industry we are not checking the reaction with the TLC. We are always checking the reaction progress in HPLC. So in HPLC, salt we are not able to identify. So a lot of problem was there in that forming the salt. So first of all, we could not identify the completion of reaction. Final product is water soluble, so we cannot do the workup also. Okay, and the purification was also a challenging task. So here purification was not possible. So what we have to purify in this stage only, because this is the organic molecule and solubility was good in solvents. But here solubility was not good in solvents since it was formed with the salt. Okay. So here also we faced a lot of problems. Still we are doing some trials to solve this issue, like to make this salt. Somehow we have made 500 gram scale by using this process only, but still we are doing this process is under progress, and we made this sodium salt. So this also how you will check. You have to check the sodium content. Then only we will come to know whether this is under specification limit or not. Okay, so like that we have to see all these parameters, and when we are going for the bulk scale batches. So say in chemistry, lab chemistry is really very simple, and that is under your control. But in plant batches that is not under your control. There are many people work in that. Okay, and lot of time and lot of money we have to spend for the any plant scale batches until it is not developed. Okay, once you set the process, everything is set, then there is no problem. But while doing this process development, you have to struggle a lot. Okay. Now, finally, the conclusion, what I understand, always expect the unexpected result. Okay, you should always be ready to solve the problem. It's not like that we are not facing this problem. It doesn't mean you will not face the problem in the pilot plant. Okay, so you should be ready to solve all the problem, clear your concept nicely, so that if you read a lot this chemistry, you will come to know, we can solve this problem like this. You will have some alternatives. Otherwise, it will be very problematic. If you don't understand what is going on, how this reaction is taking place, and what wrong things can happen in the chemistry, that you will come to know when you know the basic chemistry. Okay. Thank you. If any questions there, please ask.
I request the students, if you are having any kind of query, you can please come up with your question. Sir, I do have a question from you.
Yes.
I'll take the opportunity to ask a question from you. Sir, it's out of the league, इससे नहीं है, but अपन ग्रीन केमिस्ट्री की बात करते हैं सर, चाहे हम US EPA की बात करें, if we talk about the Royal Society of Chemistry, they are also focusing on green chemistry. But as we can see, mostly in the academic or even in the industries also, we are always focusing on the use of these kind of solvents. So
Yes,
is it easy to switch to those green solvents, or is it a challenging task for us also to use those green solvents?
Actually, madam, some of the chemistry we can use green solvents. But if you see that process is very complex. It's not about one step or two step. Okay. See, if it is one step synthesis, okay, we can think of we can use green solvents like ethyl acetate, acetone. Some of the solvents are really good. Okay, ethanol we can use, but it will not work every time. Some we can go. Obviously, we have to think about green solvents and all. But in industry, if it is possible, then okay, people will go for. They will prefer the green solvent, green chemistry only. But that area, I feel it's limited. Actually, it's not like it's not working for all the reaction. You think if you are going to do n-butyl lithium reaction, we need strong base. How we will use? We cannot use this green aspect of it. So simple chemistry like click chemistry or simple triazole rings you have to make. That kind of reaction can go in water also. Okay, we are doing, but we cannot we cannot avoid this other solvents also. In green chemistry, it's really very good thing, and it's not dangerous also for the person who are working in the pilot plant.
Yes.
So if you are getting, we are also doing. If you are getting some schemes, we can make like yes, we can use water also. We are using because the our main task is to reduce this cost. We have to whatever the process we are developing, we are also trying to develop a cost-effective approach. So green chemistry is possible, but not